All right, Mitya.
Okay, so I'm going to talk about how we can use Connectomes to learn something about computation. But before we do that, I want to start with this visual illusion. Make sure that it is shared with the other room. That's a little too fast. It's a little smoother. If you fixate on the tip of the snowboard here, then you see that the snow is coming at you down all the time. Never moves up, always moves down. But the picture never advances. Right? So you always see motion down, but there is no displacement down. Everyone sees the illusion? Yeah. Okay. So how does this work? Not yet. How does this work? So let's it's not working. Let's do it this way. Okay. Okay, so this is a looped video of 4 frames. And this is the first frame. And then watch when I make a transition to the second frame, it's shifted down. So of course you perceive that as a movement down. But the interesting thing happens from the third frame, which jumps up, but at the same time changes contrast. Okay? And so it turns out that the brain perceives a frame that jumps up, but changes contrast as the frame moving down. Then the fourth frame is again the negative moving down, so that's again movement down, and then you look back to the frame positive up, which change contrast, but it looks like down. Okay? So that's how it works. So you can go back to the PowerPoint. All right, so now just to, okay. Is it shared? Okay. So now, just to review. So you have 4 frames, and you jump down. That's of course perceived as a movement down, and that's called the pi illusion. You know, all Hollywood and film industry is based on that, so that sounds familiar. But the interesting thing is this, right? That the image jumps up, but you still perceive it as a movement down. And that's called reverse pi illusion. And then again down, and then again reverse phi, okay? So this illusions, both phi and reverse phi, are observed not only in humans, but in all visual animals where people looked. And so to understand the mechanism of how this works, we're going to focus on the simple circuits, because we think that the mechanisms are shared between all visual animals, since both illusions are the same everywhere. So our story starts about 70 years ago, when 2 German scientists, Hassenstein and Reichert, wanted to understand how animals detect motion. And they built this apparatus that they called Spangenglobus, okay? I should probably use the pointer here. So this apparatus called the Spangenglobus, it's basically, you know, 4 pieces of straw, right? Like Germany after World War II, so they didn't have any money. So they put together 4 pieces of straw, and they had a beetle whose back was glued to the apparatus. and the beetle was grasping the spanking globus with its legs and walking on it, okay? And just like mice in a maze that you heard about from Nathaniel, it could choose to go left or right at every Y junction. And they recorded how many times the beetle decided to go left or right as a function of the visual stimulus that was presented to the beetle. And what insects do, if the stimulus moves to the left, they also move to the left. If the stimulus moves to the right, they also move to the right. And they saw that the beetle experiences this phi illusion. But they also tried the reverse phi illusion by changing the contrast. And they observed that the beetle also suffers the reverse phi illusion. That is, if you change the contrast, when you move the stimulus, they perceive the opposite direction of motion. So based on these experiments, they decided to come up with the simplest possible model of the motion detector mechanism. And you probably all realize that, but just to make sure we're on the same page, the question I want to go over first is, can you detect motion with a single photoreceptor? Does anyone know? If an animal just has one photoreceptor, can it detect moving stimulus? No. Why not? Because if the object moves to the left, it abstracts the view, and you get just signal like this. And then if the object moves to the right, you get exactly the same signal. So regardless to which direction the object is moving, the animal gets the same signal. And so it cannot tell the difference between the left and the right movement. Okay? Yes?
But this even works, that if there is a stimulus that is on and off in the middle without any
Exactly.
that's the big problem, right?
Exactly. That's called the aperture problem. It's like observing the world through this really, really small peephole. And then you can't tell if the person may move this way or that way. Okay, so that's what's happened. So Hasselstein and Reichert realized that as well, and so they constructed a circuit with two photoreceptors, right? So as simple as it can be, you need two photoreceptors to detect motion that observe adjacent locations in the visual field, right? And they hypothesized that there is a delay in transmission in one of the arms, and then the inputs from the two arms get multiplied. Then, if there is an object that moves to the left, it excites the right arm first and the left arm second, but because of the time delay, the two signals arrive to the multiplication unit at the same time, get nonlinear enhance, and signal leftward motion. If the stimulus moves to the right, then the two signals arrive to the multiplication unit at different times, and they get multiplied by zeros, and then there is no signal. And of course, they can also build a mirror symmetric movement unit that detects movement to the right by putting a delay on the left arm, and then the same thing happens, and the motion is detected. Then you can combine those two units into one and add their signals with opposite signs. And then you have, this is what is called the elementary motion detector by Hasselstein and Reichert, that produces positive output when the motion is to the right, and produces negative output when the motion is to the left. The reason Hasselstein and Reichert put in a multiplication here is because they wanted to reproduce not just the phi illusion, but the reverse phi. And you can figure it out that first you blink bright stimulus here and dark stimulus there. Even though the movement was to the left, because it was the minus sign, minus combines with this minus, it gives you a plus. So this elementary motion detector also suffers reverse phi illusion. And that's why they thought that would be a good model for motion detection in insects. Any questions? Okay. So this model had a very big impact on the visual neuroscience in invertebrates, and many predictions, experimental predictions of these models has been tested over half a century in several different labs. And to give you an idea of how it's tested, I'll show you like a modern rendition of this experiment. But of course they use slightly older, yes, please.
Can you please repeat how the model replicates the inverse? It's important to the rest of the
So you play bright stimulus here and dark there, okay? So this is like a movement to the left, but with switch in contrast. Now, the bright input gets delayed here and arrives at this multiplication unit at the same time as the dark signal from here. But the dark signal is like a minus. So you multiply minus by a plus, you get minus. And then there is a minus here, so the output is a plus. And nothing goes through this arm because the signals arrive at different times. And so you get a plus, but plus corresponds to the movement to the right. So that's the reverse phi illusion.
And it was also correct to start with the black and then the white?
Okay, so in this case, yes. In this case, yes, and that corresponds also to reverse phi, like we saw in this snowboarding illusion. But things get more complicated in reality. We will see that. Okay, any other questions?
I actually have a question.
Yeah.
Don't people use a circuit like this for intraoral time delays also?
Also, yes.
Is there a similar illusion in sound localization?
So that's a good question. So there are similar kind of illusions with frequencies that I've heard about. I don't know if there is also one with sound localization. Any other questions? Okay, so to test this kind of model, people did experiments that evaluated motion detection in two ways. One way would be as shown here, you measure the rotation of this ball because the fly started trying to turn in the direction of the stimulus that is presented to it at the same time, so it's trying to follow this direction. And it rotates to the right, as shown here with the black line, and when the direction of the movement switches, then the fly starts turning to the left, and you will see that black line will go up. So that's one way to assess the motion detector through behavioral output. And another way is they found this giant neuron called HS, or in big flies it's called H1, that you might have heard about, and you record calcium activity in it, shown in green, and you can see that that neuron responds when the movement is to the left. So this is like a motion detection neuron, HS, and it's so big that it can be recorded from electrophysiologically or using calcium imaging. And so these are the two ways that people tested the prediction of Hasselstein-Reichert model through behavior or through the activity of the neuron HS. And all the experimental results confirm the predictions of the Hasselstein and Reichert model. And so it became a canonical model of neural computation, and people have even argued that a similar model can apply to vertebrates, and those arguments are in fact reasonable. So you would think that the story is over here, but it's just the beginning. And the reason is that all these experimental tests we're testing the mechanism of motion detection indirectly. And to explain what I mean by indirectly, I have to tell you a little bit more about the fly visual system. So just the necessary stuff that you need to know, okay? So you know that the fly eye doesn't have one lens like ours, but it has many micro lenses, 800 or so. And there are photoreceptors under each micro lens, and those photoreceptors send luminance signals downstream along these processing layers that have these names that are not important, lamina, amygdala, lobular plate. And we know from physiology that the information from the photoreceptors is initially processed in a segregated manner, where information only from one pixel is processed in the first layer of lamina. And as I told you, a single photoreceptor cannot detect direction of motion. So there's no directional selectivity here. But then, nearby channels start exchanging the information in the medulla, and we saw in this calcium imaging that this neuron HS, which is one of these colored neurons, actually responds to motion. But these neurons do not compute motion themselves, they just integrate motion signal over the visual field. And the cells that first compute motion are known under the name T4. They come in 4 subtypes, which correspond to the 4 directions of motion, north, west, east, and south. And 4 of them appear 800 times under all those pixels. So they compute the motion signal locally and then transmit it down to those integrated cells from which people recorded it. Of course, the behavioral output is also driven by these giant cells. And so this is why I told you that those experiments that tested Hasselstein and Reichert were indirect, because they only recorded what happened either in those integrating cells or in those in the behavior driven by those integrated cells. So people hypothesized that elementary motion detection, those Hasselstein-Reichert units, actually took place in the medulla, and the T4 was computing this Hasselstein-Reichert motion signal. But this was just a hypothesis, and nobody could test whether it's correct or not, because nobody could record from those cells physiologically. And the circuitry was not known in the medulla. And the medulla was the terra incognita. And people just hypothesized that that's where Hasselstein-Reichert motion detectors were. So when I decided to initiate connectomics, I thought that this would be a great target to reconstruct and to find the circuitry underlying the motion detector. And I had no issue. I mean, Hasselstein-Reichert seemed like good guys, so, you know, we were determined to find the Hasselstein-Reichert motion detector. But to find it, of course, is very difficult, because no one has done a reconstruction of this complexity. And so, you know, this is a cartoon that shows how connectomics is done. You know, you take a piece of the brain, you slice it into thin sections, like prosciutto, except 1,000 times thinner. And then you image each slice under electron microscope, and then you get this grayscale images where contrast comes because there are a lot of labeled proteins in the membranes. And then you segment those images as shown here by false color, and the same color is supposed to correspond to the sections of the same neuron, so then when you put them back together, you can reconstruct in 3D, identify synapses, and then trace from those synapses to cell bodies to reconstruct the connectome. So perhaps looking at this cartoon, you think that this is a really simple task, We walked in the park, and I was misled to thinking that it was simpler than I thought. And so, the first thing we realized is that there are so many photographs that you cannot trace them manually. There are millions of photographs from electron microscope. So we had to train a computer vision system to do that. And to train the computer vision system, I had to learn machine learning, which in the end turned out to be a good thing. Anyway, 7 years later, we were able to reconstruct the medalla circuitry that we were interested in. So out of the raw data emerges the reconstruction of those 3D shapes of neurons corresponding just to a fragment of the medalla, to 7 adjacent pixels, or columns as they're called. These are all the membranes, all the neurons in that volume. So basically, we, in addition, of course, to volume metric reconstructions of neurons, we also identified synapses, and so we got the full connectome of this region with all the neurons and all the synapses, but I'm not going to, show you the whole connectome. I'll spare you the trouble and show just the part that is relevant for motion detection. And it turned out to be amazingly simple. Of course, it's an oversimplification. It's a cartoon. I'll show you more real data later, but that's the cartoon you need to know to understand the rest of the talk. So you can already see that this is a little different from Hasselstein-Reichert motion detector. Okay, so the main difference is that Hasselstein-Reichert said, oh, you can't detect motion from a single photoreceptor, we're going to detect it from a pair. And that you can do. But in the real system, you need inputs from at least 3 pixels to detect motion in this neuron T4. And that was the first surprise. Do you have a question? There is a slight variation in terms of how the delays are implemented, but that's actually not important. There was just this channel that computed a derivative, as opposed to the other channels that were low-pass filters. But that's not very different from the time delays, because even before we got into this business, people have pointed out that the biologically plausible way to implement time delays is through low-pass filtering. So they actually did build those Hasselstein-Reichert detectors with low-pass on one arm and high-pass, which is like a derivative on the other arm. So that wasn't that big deal, but the interesting thing is that it was 3 pixels, and they come in with all this signs with pluses and minuses. So observing this suggested that the Hasselstein-Reichert model needs to be revised. Any questions so far?
So, the derivative of the signal be calculated in what way? So how the derivative of the signal be calculated?
Right, so the derivative signal, can I move this? So the derivative signal was calculated in the following way. You know that for a neuron, you can measure by reverse correlation, you can measure response of neuron as a function of time. And the temporal filter that describes this channel was like this. So it took this with a plus, it took this with a minus. So this computes a temporal derivative. And that's what happens with this channel.
Okay, so that is similar with time delay.
Exactly. So the other channel was like this. So this was like the proportional channel, and this was the derivative channel. Okay? And that is recorded in physiology. I'll show data later.
Yeah, I was going to ask what part of this comes from physiology and what from the connectome?
So from the connectome, we have the circuit and the identification of these channels, because they correspond to different cell types that repeat in every pixel. So in every pixel, there are these 3 cell types, and the 3 different cell types in every column, but only one of them makes a synapse on this T4. And then T4 dendrites have this shape. They're like this, for the T4 that responds to this direction, and then the inputs from 3 pixel, they make synapses on this dendrite. Like this. So we could find both the fact that they come from 3 adjacent pixels, but also in specific ways. So these are called MI9, MI4, I think, and this is called MI1. So these are different cell types, and each of the cell types is present in each pixel. But this neuron only grabs this one from the middle pixel, this one from the right, and this one from the left. And when we looked at the neuron that responded to the motion to the other side, its dendrites were like this, and then it would grab MI9 from this, and MI1 from this, and MI4 from that.
The positive and negative sign of this?
The positive or negative we could not get from connectomics. This was obtained later by both doing RNA-seq identification of neurotransmitters, and later they figured out a way how to actually extract the data from electron microscopy through actually deep learning. It's an interesting story, but I'm not going to deviate, but we didn't know that at the time. So now we know for sure what the signs are. Okay, any other questions?
Yes.
Yeah.
So the T4 neuron can just detect 2D.
Can detect what?
2D motion.
One neuron only detects motion in one direction, but there are four neurons, and they correspond to the 4 directions of motion.
That cannot correspond to back forward and back.
So in the fly, the eyes are on the side, and so this 4 sides is up, down, forward, and backward. Okay?
I have another question.
Yeah.
There are 3 neurons that is the input, the T4 neurons.
Yes.
I think the most important one was the derivative neuron. So there are 2 other neurons that
Yes.
seem to convey the identical information of the original information. So are they necessary for this motion detection?
Okay, that's a very good question. So as we discussed, if there was just this input, it would be 1 photoreceptor, you could not detect motion direction. But if you add 1 more, that's enough. And that's Hassenstein-Reichert. So 2 are necessary. Why there is a 3rd will be a plot of, that's why I'm giving a talk. Okay? All right, let's move on. So so how does this circuit, so we just covered how Hassenstein-Reichert detector detects motion. How does this 3-pixel circuit can detect motion? So, let's go back to the basics and think about what motion detection requires by observing, sort of think about observing sequences of visual frames, right? So you see a photograph, like that snowboard, and then you see a shifted photograph, right? So let's look at some region of that photograph and plot pixel luminance as a function of space for that object. And there is some, border of the snow or something, right? Then at next time point, the object itself does not change, right? It's invariable. But it shifts to the right. And that would be a motion signal, right? So how can we detect the motion speed from these 2 frames of the video? Okay, to do this, we can find a relationship between the displacement, which is just given by the motion speed for unit time, the temporal change in the luminance, which is this delta x with the subscript t, and those two can be used to compute the spatial derivative of the luminance profile, right? So that's just the definition of the derivative. So the spatial derivative of the luminance, right? The tangent is given by the ratio of the temporal change in the luminance divided by the speed, which is displacement in unit time. So that's just a geometrical relationship. Now, if we were able to measure the spatial derivative of the luminance in the image, and we could measure the temporal change, then by knowing these two things, we can find the speed. That's the idea. Okay? So this is how you can establish the speed of the movement of a moving image from two frames. But this is, so you can say theta is the temporal change divided by the spatial derivative. But dividing is not a good idea in real life, especially derivatives, because they're very noisy. So instead of dividing, what do you do? Well, you solve an optimization problem, which is minimized when theta is given by this, okay? But you penalize for the deviation from that optimal solution. And also, you introduce this regularizer on the speed, which favors lower speeds, just like in ridge regression. If you like Bayesian approach, you can think of this as being log likelihood, right, for Gaussian signals, and this is log prior. So that you have a prior that velocity is unlikely to be too big, and then the signal and the noise is distributed Gaussian, and so you minimize this with respect to theta. Okay? So now if you do this in the low signal to noise regime, it turns out that this is the ridge regression expression, but then you can ignore this term when the noise is very, very big. And then the speed is just proportional to the correlation between the temporal and spatial derivative. Of course, this only works in low signal to noise regime, and it only works on the signals that are normalized to luminance and contrast normalized images, right? Because if I increase x, scale x up by a factor of 2 because, you know, the sun is brighter, then this will increase by a factor of 4, and I will think that the velocity is 4 times higher. That's not a good motion detector. So what happens in the actual circuit, and I don't have the time to go into that, before signals get to the T4 cell that detects motion, there is another layer of cells that does normalization to luminance and to contrast. So the signals that T4 cells see are the normalized contrast signals. That's why this method can work. Now, let's try to map it back onto the circuit we found from Connectomes. And as I told you, we need to compute the velocity by multiplying the temporal derivative by the spatial derivative. The temporal derivative is computed by this channel here because the cell that provides this channel takes the derivative already. And the spatial derivative, of course, we can compute by just taking the difference between those channels, and to make it centered onto the derivative channel, we compute the difference between this, and that's why you need opposite signs here. Okay, so this plus here and the minus here, they compute spatial derivative, and this input computes temporal derivative, and if only we could have a multiplication in this neuron, we would be computing the motion detector.
So the experiments are typically done in high signal to noise ratio, right?
So this is questionable. There is an argument that they're always low signal to noise. but I can,
So what's the
so there is a paper where they try to find this regime. So to step back, the history was a paper by Pointers and Bell that said that the computation should be different in high signal to noise and low signal to noise. Okay? And in low signal to noise, they said it should be Hasselstein-Reichert multiplication, and high signal to noise should be division. Then I think it was Borst and Deck tried to change the signal to noise to see the transition between the two. And they found that no matter what the signal to noise was, it was always Hasselstein-Reichert prediction. At least that's what has been done. Now, how you interpret this is up to you, but that's the facts.
At some point, the distance between two images can be so large that you need to observe it as moving or
Right. So this is all designed to obtain movements from one pixel to the next, but over very short time periods. So that's how you deal with it. You allow small displacements only, but then you can compute the difference over short time scales, because it's all proportional to velocity.
Can the Connectomes tell you something about the maximum speed the animal can?
Well, together with physiology, yes, because we know that there's only two adjacent pixels, and if we measure temporal response of these neurons, then we can tell you what it is. But all the temporal things are still being sorted out in these circuits. So I'm not going to defend the timescales very robustly, because they're actually arguing among themselves.
So the prediction, I don't remember, I know we expect, but I don't remember the result. So the prediction is that this is in, the division is in high signal to noise ratio or low signal?
So in high signal to noise, you take the difference between the temporal derivative and the spatial derivative, which is like how computer scientists would do it for, real images, like for artificial images, right? At low signal to noise, it's a Hasselstein-Reichert multiplication that's shown here, which we replace just by the multiplication of derivatives, which is yet another way to do it.
Okay, now the other thing is, I thought experimentally you do see the quadratic dependence on contrast.
Exactly, that's a prediction of Hasselstein-Reichert.
But you said here there's presynaptic cells that normalize.
Well, I mean, how do you normalize? You have a sigmoid, and so when it's high, you get it normalized.
I see. So
But at low, you can always get to the low regime where things are linear, right? Like, okay? Any other questions? Okay. So basically, now we can map this onto the circuit, and then we can say, okay, these two inputs compute the spatial derivative. If only you could multiply, you would compute the motion signal. Let's just check that this circuit satisfies the find the reverse phi illusion before we go on, right? Because that's what the model has to satisfy, right? Hasselstein-Reichert did, so should we. So the way it works is that if you flash a bright stimulus over this pixel, and then later flash the bright pixel over the central pixel, then because there is a low-pass filtering, there is a coincidence between the input from the left pixel and the central pixel, and then you have two positive inputs, they add up or multiply, they produce rightward motion signal. Similarly, this circuit suffers reverse phi illusion. For reverse phi in this case, you need to flash, that's why I answered the question before. It's really important that you flash a dark stimulus here and the bright stimulus in the central pixel. Then, because the dark pixel is low pass filtered, it coincides with the bright onset, and it also produces the right stimulus because of that, the right motion because of that minus sign. Okay, so the dark stimulus gets multiplied by it's a minus, gets multiplied by the minus, it's a plus, then gets multiplied by the plus, that's also positive, which is motion to the right. So this experiment has actually been done by the Clark lab, and they found exactly by shining a laser on this pixel and then a laser on that pixel, that this neuron responds with the phi and reverse phi illusion. And by the way, the thing doesn't work if you simulate this 2 pixels for phi or this 2 pixels for reverse phi, which kind of supports the specificity, the anatomical specificity that we found. Okay? But you may still ask, what's the mechanism of multiplication here? And that has been like the holy grail of this field, and it is still unclear whether multiplication really takes place in the dendrites. Some papers tell you that the summation in dendrites is linear. Some papers argue for nonlinear summation. Now, in general, it may not be such a big deal for this model because, you can have a nonlinearity in the synaptic transmission downstream, right? Because there is a rectification, so there is a nonlinearity downstream of this linear summation. But from my perspective, I don't think there is a clear answer whether there is a multiplication or not in the dendrites. And by the way, I should mention that at least one of these experiments or models has to do with the mouse, and it's thought that a similar mechanism is invoked in directionally selective starburst amacrine cells in the vertebrate retina. So in that sense, it's a convergent evolution. Okay, so we have a new model now, this 3-pixel derivative correlation motion detection that is confirmed by anatomy and supported by physiology. But there is this half a century of all other experiments that confirm the predictions of Hasselstein-Reichert model, one of which Rafa mentioned, which is this quadratic dependence on the contrast. Okay? So perhaps this one we can reproduce in our model, but there are many others, the frequency dependence, the velocity, and so on. They're all agreed with Hasselstein-Reichert. So what about us? Can we explain all those things? And it turns out, and it wasn't obvious for us in the beginning, but we realized that although the two kind of circuits, the Hasselstein-Reichert on the left, okay, I redrew it a little bit differently, and our derivative correlator where I showed the computation of the derivative explicitly to facilitate comparisons, they look like two different circuits. But once you add all the ones that are next to each other under different pixels that are being summed in this large neurons called HS, the calcium imaging of which I showed in the beginning, then the two circuits become the same. So after integration, there is no difference between Hasselstein-Reichert and derivative correlation. It's not obvious from this figure, but I'll try to show it to you by doing this little trick. So if you look, for example, at this two blue channels, okay? So you will see that it takes the input from the left pixel, and then it multiplies by the input from the right pixel with a minus sign, and then there is a minus sign on the product. So together it's a plus sign. And then look at these green channels. It multiplies again the left pixel with the minus sign by the right pixel, and then there is no second minus sign. So this is the same product of the luminance detected by the two pixels, but with opposite signs. And then they are summed together in the big cell HS. So these two contributions cancel. And what you're left with is this. And now you can see that this part looks exactly like the Hasselstein-Reichert motion. You can do this algebraically as well, and then you find that once you sum the inputs of the Hasselstein-Reichert detectors and or the derivative correlation detectors over the whole visual field, you get the same answer. So therefore, our model is validated by all the same experiments that were done over half a century to test Hasselstein-Reichert. Okay? Any questions?
Why create this additional problem?
Exactly. Thank you. So even though the integrated signals are equivalent, we find that the primitive, computational primitive here is very different from the one that Hasselstein-Reichert suggested. And as a physicist, I would always favor a solution that is simpler, and 2 pixels is obviously simpler than 3 pixels. And you can't go to 1 because it doesn't detect motion, so why have 3 instead of 2 when you can have 2? This seems ridiculous. So anyone have any suggestions of why you would want a 3-pixel motion detector? Why wouldn't you be happy with the Hasselstein-Reichert? Yes.
Maybe if one of the tails, the others can compensate.
Okay, robustness, graceful degradation. That's one idea. Any other ideas?
Could be the distance. So if you, it's different if you just move the directly left or near right. So.
Right. So that's a possibility also that it's more tolerant to different velocities of motion. Any other ideas?
So it's an idea formulated as a question. So is there a difference in how they process 3-point correlations?
Probably yes, but that's complicated because you might think that in the Hasselstein-Reichert 3-point correlations don't exist, but they don't exist in space, but they exist in time. So you're asking about spatial correlations? Yeah, then certainly there could be a difference. Yeah, that's a good point.
Is to resolve any other ambiguity that you can have about the natural stimuli. Like I'm thinking of like something that flickers in the center and shadows laterally, like in 3 pixels. Yeah.
Okay.
Like what Rabbi was saying, like some kind of. Is to disambiguate anything that is obvious that another stimulus.
Yeah, so that's the right direction. So it's in the direction of natural stimuli, right? We want this to detect natural stimuli. But what is it? How do we know it's a natural stimuli? How do we adapt to it? Okay, so I'll open the cards. So the answer is that you can, if your goal is just to build something that detects motion, Hasselstein-Reichert detector is fine. Okay? But if you want the detector to be adaptable or learnable, that's when you need a 3rd pixel. Okay? So why do we want adaptable and learnable? Well, because we want to adapt to natural stimuli, we want it to be robust about computation, but you can say, oh, but flies, they're cheap, you just make a lot of offsprings and the best ones survive, and anyways, the circuit is determined by the genes, so why do you need to rely on learner? So this argument are not completely silly, and the general architecture, of course, is laid by the genes, but I believe that there is a learning in terms of activity-dependent synaptic plasticity there, and I'll try to give you an argument of why I think so from our connectomics results. Okay, so by the way, this is the reverse correlation of those 3 channels that I was doing on the board. So this is actual data from calcium imaging from the Bourse lab, and so the central channel is blue, and there's a red and green side channels. And the reason there is a minus here is because I wanted it to be consistent. So because there is a flip here, which is red channel flips, the true synapses in the real connectome is inhibitory here, and excitatory here. Okay, but I just didn't want to go into that detail before, but because there is a flip, I did that times minus gives plus. Okay, anyway, but these are the 2 inhibitory channels, and this is excitatory. And that's why multiplication actually makes sense, right? Because you can have shunted inhibition. Anyways, so why should there be learning? Okay, So, as I told you, this is a cartoon. This is an oversimplification of the connectome. What we actually got from the data are not just the synaptic connections, but the weights of individual synaptic connections from all the neurons. And we estimated the weights by counting the number of synaptic contacts in parallel, which are approximately the same size and fly. And so roughly, they're of the same weight. So you just count the number of synapses and you get those numbers. Now, this is the view of the fly. If you like stare at the fly eye from the top, then you see this honeycomb lattice, okay? And these are individual pixels. And these are the numbers of the number of synaptic inputs, the weights of connections onto this T4 cell that responsible for the motion to the right from the central MI1 cell, from the blue channel. It also turns out that nearby pixels also provide little inputs, so it's kind of like a Gaussian a little bit, okay? But an order of magnitude smaller, so that's why I have this cartoon, that most of the input comes from the central pixel. Now the green channel comes to the right, so 1 pixel off, and the red channel comes into the left. But there are all these numbers, and you can see that it's a really tricky problem for the fly to wire up, because the motion detectors, they have 4-fold symmetry. And the lattice here is honeycomb lattice, it has a 6-fold symmetry. So all those numbers are different for every T4 cell, and there are 800 cells responsible to the motion to the right, and then there are 800 cells of each other of the 3 categories. And every time you have slightly different numbers. So that means that you need to remember 10 numbers for each T4 cell, and then there are 3,200 T4 cells, so then you need to remember 32,000 numbers. Okay? So try to encode that all in the genes. Right? You will say, oh, you don't encode the numbers, you encode the rules. Exactly. So these are the learning rules that are implemented by the synapses, we think, and they learn and fine-tune the circuit to detect the motion the best way. So that's my argument for why we think that there should be learning. Okay? But why does learning account for the 3rd pixel? And this has to do with something that you already heard about at Nienlong's and before, that you need error signal to learn, right? If you don't make any mistakes, you never learn in life. The same is true for neurons, right? You need to have an error signal all the time. How can you get an error signal? So it has a Steinreiter detector. All it does is detect motion. And you need the minimum of 2 pixels to detect motion. So once you detected motion, where will the error come from? Okay? So the error can come if you make a prediction, right? So if I can predict something and then see the real thing, then I can have an error. But I cannot predict the direction of motion from 1 pixel. So I need 2 to make a prediction. Okay, but how am I going to test this prediction? I'm not recording anything from this pixel. And that's why the 3-pixel motion detection makes sense. You can detect motion with the first 2 pixels, make a prediction for the 3rd, and then subtract with the minus the actual observation. So that's why we think you need 3-pixel motion detector. So you don't just detect motion, but you can learn the motion detector. Any questions?
The dense enough network would always have 2 pixels on the same object. So there's 2 detectors. If you have 2 detectors that are close enough in space, they detect the same object. There would never be 1 pixel size object moving the eye.
So think of this, I oversimplify this because I have to, but the real thing, what happens is that it detects an edge of a moving object. So think of this as an advancing edge. And first there is an increase in the light intensity here, and then there is an increase here, so it takes derivative, and that's why they trigger together, because it's an edge advance. And then you predict that that edge continues moving to the right. And that's your prediction for this pixel, and then you subtract the actual observation. That's what you think is going on.
Where does the prediction comes from?
How does the prediction comes to the third neuron?
There is no third neuron. Everything is computed in T4. And the way I'm breaking it down for you isn't how it happens mechanistically. Okay, so we'll do the mechanistic, well, I guess I'm running out of time. I was going to do the mechanistic description next to show you how this actually happens. But this is just a logical argument for why you need a third pixel. Okay?
So if they knock out some gene that is like that will make those inhibitory neurons disabled, what should we get? I mean, flies cannot see, understand emotion.
So those experiments have actually been done, and it was shown that these channels are essential. But when they have been done, we did not have this prediction error perspective. So we need now to go back and re-examine that, maybe even redo the experiments, I don't know. But that's a very good question. So this is actually in the fly because there is a way to target cell types of each of those three inputs individually and specifically. Those inputs could be indeed selectively silenced.
I see.
And they have done these experiments. But I need to go back to re-examine them because when they did it, they didn't think about it.
Yeah. The other thing is that in evolution we have a lot of pretty silly things. So why don't we take, like, as an explanation that, oh, it's just an evolution, that's why there are 3 neurons there.
Right, but then we don't have to do theory, right? Everything is a historical accident, yeah?
Okay, thank you.
So are there experiments on learning in this context?
So that's a really good question, and again, there is controversy there, and these are pretty old experiments, but to summarize them very briefly, okay, they try to show that this circuit is built according by the genes, by raising flies in the dark and then bringing them into the experimental apparatus in light, and then they see motion. Of course, this doesn't prove anything because there could be a spontaneous activity, just like retinal waves in mice, that could build it up. Also, the detector may not be fine-tuned when they first did this with high contrast, whatever. But there are other experiments that show that if you count synapses, those numbers that I showed you, they vary by at least 10% every day, from day to night. There's a 24 cycle, right, a circadian rhythm, where the number of synapses changes by at least 10%. So the system is incredibly plastic, and it constantly adapts. Now, so there is evidence of adaptation, there is evidence of the role of the genes, but what we don't know is what are the learning rules that lay down the circuit. That has not been established.
For example, if you had a stimulus with only phi type motion or reverse phi type motion, would you expect that they would learn kind of?
So, okay, so that's a good question. I should emphasize, and that was the point about natural images. In natural images, there will never be a reverse phi. Right? Reverse phi is a natural input that was artificially created to produce an optical illusion, right? So even though the flies in evolution have never seen reverse phi, they're subject to that illusion. And I think that's the tip-off of this prediction error mechanism. So that is a liability of this prediction error mechanism. But the flies don't suffer from it because it never happens in natural world. There was another question, I think.
When I was trying to connect this circuit with the speed estimation formula,
Yeah.
we'll take multiplication of the derivative signal with the difference of the signals. I'm trying to think about where is the prediction part. So can I break apart the formula like the left cell times the middle cell minus the right cell times the middle cell?
Yes, exactly.
Okay, and the former part is like a prediction.
Exactly. Exactly. Well, okay, I'm running out of time. What do the bosses tell me to do?
1 more minute.
Okay. Any more questions? Okay. So what we did, we built a model that tried to emulate learning. And I don't think I can explain how it works in 1 minute. So I'll just claim that you can build a model based on the smoothness of transformation using something called Lie group theory, but it's not really Lie group theory, it sounds scary. It's a very simple linear approximation to the change in the image luminance that is a linear function of the stimulus itself times this matrix that is called the generator, and theta, which is the speed, okay? And if we knew theta, we could evaluate A by linear regression, By linear regression, we could just evaluate. But we don't know theta. So what we have to do is something like principal component analysis, where we have to evaluate theta and A at the same time, or clustering, and we can do it, but we have to use this outer product features, which is the time derivative of the input times the input itself, okay? And if we use this outer product features, because these are vectors over pixels, then we can actually evaluate A, and once we have A from PCR clustering, we can detect theta by sandwiching this A between X. So if you want to know more, you can ask me, but this A can be learned in unsupervised fashion from paired sequences of frames that are shifted, and you get this kind of pattern of, plus and minus inputs, which correspond to the circuit, right? So you can map this on, wrong way. We can map it on the circuit where 1T4 implements one row of the generator, where this is the plus, this is, the derivative comes here, and that's the row, and then the other column is this minus, okay? And so, because there are many T4s, they all combine in this large tangential neurons from which calcium signal was recorded from one direction or the other direction, and to show that our learning algorithm can learn not just translation, but also rotation, which is crucial for flies, right? Because when you fly, there's a lot of rotational motions around a different axis, and so the optic flow is like this, and we can learn from rotation pairs of rotated images. We can learn the detectors like the same way we did before. This would be inputs to T4s. Sorry, I have to rush. And one last thing I want to say is that why do you need to know about motion detection if you don't care about flies or motion detection? Well, that's because I think that is the simplest example of learning content invariant transformations. And learning content invariant transformations are crucial to things like pattern recognition, right? If you want to recognize spaces belong to different people, these are two different people, this is one person, this is the other person, okay? But the pixel level similarity between those pixels is better between the same view, the same side view of different people than the different side view of the same person, okay? So how do you learn to detect the same face regardless of which direction you look at it? And for that, you need to learn this transformation that keeps the identity of the person invariant. And I think that if we were to learn how it's done for motion detection, for translation, then we can use it to learn more abstract invariances, and that is potentially a biologically inspired way to build artificial intelligence, okay? So this leads us to the summary. So what we did is that our connectomics results, they falsified the constant side record detector in favor of this 3-pixel derivative correlator motion detector, which, you know, when integrated over the whole visual field, gives you the same answer as constant side record, but it led us to think why you need to have 3 pixels, and that led us to learning. And we think that if we understand unsupervised learning or motion detection, it will pave the path to biologically inspired AI for more abstract computations. And finally, the acknowledgements. Connectomics was a big effort, both in my group and the collaborators, and these are the 2 key people who worked on the learning theory of motion detection. Thank you very much.
Any other questions? Yes.
Can a fly detect steps? That's information.
Detect steps?
Yeah, like our humans so I to detect them.
Oh, depth, depth. Yeah, so the parallaxes, right, because you see different views from different eyes. So that's a good question. Most animals, they have very small binocular overlap. And so most of their vision is monocular, but there is a sliver of the central visual field where the visual fields overlap between the 2 eyes. So they can perhaps do that. And in fact, it's known that the male flies use that kind of overlap when they chase female flies in flight. Okay? Now, the mechanism of that, I don't know. So I don't know what it is based on. But at least there is a part of the visual field where there is a binocular overlap. So that could, in fact, be done.
Hi. Does the model take into consideration auditory information if the moving object is also making sound, and we have sound localization? Does that add to the information to the same model, or is it different?
So there is multimodal integration in flies, of course. It probably doesn't happen on the level of T4s. It probably happens downstream. I mean, certainly one place where it is known to happen is the central complex, where, you know, 2 synapses downstream from this big integrated tangential cells, they get to the central complex. That's like, you know, when they determine the heading direction. So there, they definitely have multimodal integration, but I doubt it happens at the level of T4s.
Cool, thanks.
All right, if there's no more questions. Okay, so I want to ask if the unsupervised learning only happens during development, or it keeps changing size during the experiment. So, yeah.
That's a good question. I don't know, but I would so in the mammalian brain, where there is more work on synaptic plasticity like LTP, it is known that some circuits are only plastic in development, while other circuits stay plastic throughout the whole life. And I don't know if there is correspondent work in flies.
All right, that's that.
Thank you.